and welcome to Malvern Panalytical's web seminar program. My name is Birgit Toscher and I will be your host for today. The webinar today is entitled Biologics Masterclass Immunotherapy Discovery and Development. Our presenter today is Dr. Justyna Czepecka. She is Principal Scientist for Immunology. And Justyna first joined Aquila, now part of CLS, in 2014 as senior scientist, following completion of her PhD in immune regulation at the University of Warsaw and the University of Humboldt, and then several postdoctoral roles exploring the therapeutic potential of small molecule drug candidates against allergic disorders and autoimmune diseases. Throughout her academic career, she authored or co-authored over 20 publications in peer-reviewed journals. And she gained further industrial experience with antibody analytics as an associate director before returning to CLS in 2019 as principal scientist for immunology. And Justyna is leading a scientific team designing and executing novel and tailored client-led programs in immunology and immuno-oncology research. And before I hand over the word to you, Justyna, I would like to welcome you all for participating in today's webinar, and I would like to inform you that this is an active program and that we would like to encourage you to ask your questions anytime you have them. Please type them in the question and answer section on the right side of your screen. If you don't see this question and answer section, then please click on the three dots which you will find on the right bottom corner of your screen, and then the question and answer section you can pull up. And another feature, interactive feature, which we will have in this webinar is a poll. And this poll will help us to understand our audience in this webinar and helps us to improve further webinars. And I will open the poll now, and we would very much appreciate if you can take your time to answer the questions. Yes, this will help us very much. So let me get the poll questions open to you, and you should see it. You will have to complete presentation. This webinar will be recorded, and you will find it within a few days on our website in the On-Demand Knowledge Center. And for those who listen to this on demand, please send your questions to events@malvernpanalytical.com. The questions which you will type in will be read loud by Justyna after her presentation, and she will give you the answer. If we run out of time, then please be sure that all questions are captured and that we get back to you personally. And this is so far from my side. And now I would very much welcome you, Justyna, for your time to take this webinar with us through your webinar. And now please start with your presentation. Thank you.
Thank you very much, Birgit, and hello to all the participants. Thank you for dialing in. We'll really appreciate your interest. So during my talk today, I would like to present to you our capabilities and solutions we provide within the immunotherapy drug discovery space, with a specific focus on two types of in vitro platform assays, one that models T-cell exhaustion and the other that models tumor-associated macrophages. These two types of Cells are major players in the tumor microenvironment, and not surprisingly, they also constitute major cellular targets in immunotherapy. I will show how we have established robust laboratory assays using human T cells and macrophages, and how we can use these assays to identify promising compounds that would benefit cancer patients. So before I dive into the main part of my talk, I would like to tell you very briefly about who we are and what we do. So Concept Life Sciences is a contract research organization offering a comprehensive set of services in the field of drug discovery and development. Our team of immunology experts specializes in designing, developing, and conducting in vitro and ex vivo assays using human primary immune cells. These assays are often complex, multicellular culture systems that closely recapitulate the different cancer settings. We have a consultative approach to the services we provide, and we can tailor the experimental design based on the target and the scientific questions that our customers come to us with. So whilst we do have some standard and off-the-shelf assays, a large part of what we do is customized or novel assay design using our in-house expertise. Our assays can be used to address multiple scientific questions, including those relating to elucidation of disease pathology, exploratory biology, target validation, compound screening, in-depth compound profiling, and most frequently in identifying mechanism of action of given compounds. Our assays involve all types of immune cells playing crucial roles in the tumor microenvironment, and we are experienced working with a wide selection of intracellular and extracellular molecular targets that are expressed by different types of immune cells as well as cancer cells. Obviously, those are essential in our assays. Our assays can be used for testing not only small molecules or different types of engineered monoclonal antibodies, but also more and more frequently by novel approaches, including cell and gene therapy. The starting point for most of our assays is through immune cell isolation from peripheral blood mononuclear cells, which we obtained from healthy donors in the form of leukopaks, Buffy coats, or whole fresh blood. Captured here on the slide is a selection of our core assays, which are broadly grouped by cell population of interest. So we have what we call T-cell centric assays, we have myeloid cell centric assays, and natural killer cell centric assays. So during my talk today, I would like to focus on one of the T-cell centric assays, that is the exhausted T-cell assays, and two macrophage centric assays, which is the reprogramming of M2 like macrophages and reversal of M2 mediated T-cell suppression. As the main part of my presentation today will be exhausted T cells and tumor-associated macrophages, I would like to briefly describe in what types of tumor these two cell populations are found and why targeting them is important in developing new and improving the existing immunotherapies. And the easiest to do it is by using the binary classification of tumor types based on the type of immune cells they are infiltrated by. So tumors can be divided into cold and hot tumors. So starting with hot tumors first, as they are more relevant in the context of immunotherapy approaches already approved for clinical use. Hot tumors are those tumors that show signs of inflammation, meaning that the tumor has already been infiltrated by T cells and natural killer cells, these two potent types of immune cells capable of directly killing cancer cells. So the tumor cells in hot tumors have undergone already many mutations that created neoantigens recognized by the T cells. So for this reason, hot tumors typically respond well to immunotherapy treatment using checkpoint inhibitors like the anti-PD-1 blockades. Interestingly, however, T cells found in tumor masses are of certain phenotypic and functional state that is fundamentally different from conventional T cells, and the state is called T cell exhaustion, and the T cells are called exhausted T cells. These T cells, amongst other molecules, express high and sustained levels of PD-1, which is a major immunotherapy target. Only a few types of cancers are considered to be hot, including melanoma, and not every hot tumor in every patient, unfortunately, will be responsive to such treatments. In contrast, when we look at cold tumors, these are the non-immunogenic tumors that have not yet been infiltrated with T cells, a sign that the immune response is not working for one reason or another. The lack of T cells makes it difficult to provoke an immune response with immunotherapy drugs. One of the reasons why cold tumors lack T cells is the presence of suppressive immune cells, such as tumor-associated macrophages, which are known to dampen down the immune response and inhibit T cells trying to move into the tumor where they can perform their cytotoxic function. So on one hand, we have the challenge to robustly reinvigorate T cells in hot tumors. And on the other hand, we have a challenge to first alleviate T cell suppression through elimination or reprogramming of suppressive immune cells before attempting to boost T cell activation through checkpoint immunotherapy. I would like to now expand on immune checkpoint molecules. We are all familiar with the first checkpoint described, CTLA-4, which was followed shortly by PD-1, which I mentioned already on my previous slides. There are, however, more checkpoint molecules, including LAG-3 and TIM-3. So checkpoints are a normal part of the immune system. Their role is to prevent an immune response from being so strong that it destroys healthy cells in the body. So checkpoints are expressed predominantly on T cells, as you can see on the cartoons, and recognize and bind to partner proteins, such as it is in the case of PD-1, PD-L1 or PD-L2, as it is in the case of CTLA-4, CD80 or CD86. For LAG-3, we have MHC2 ligand, and for TIM-3, we have galectin-9. So when the checkpoint and their partner proteins bind together, they send an off signal to the T cells, and this can prevent the immune system from destroying cancer cells. So immunotherapy drugs called immune checkpoint inhibitors work by blocking checkpoints proteins from binding with their partner proteins, and this prevents the off signal from being sent, allowing the T cells to kill cancer cells. Checkpoint molecules are overexpressed on exhausted T cells, and upon ligation, they prevent exhausted T cells from effectively killing cancer cells. So let's focus now on the exhausted T cells and why they are important in the context of cancer. T cell exhaustion is a broad term used to describe T cells dysfunction resulting from chronic stimulation. When the immune system encounters a pathogen, it mounts T cell responses. When the immune system then becomes activated, T cells start proliferating and expressing an array of molecules that enables them to fight the invader. At the same time, however, as a control mechanism preventing their overactivation, T cells also start expressing inhibitory receptors that I mentioned on the slide before, the checkpoint molecules, that is PD-1, LAG3, and TIM3. And when the pathogen is eliminated, T cell responses decline over time, but some T cells survive and create a pool of memory T cells being able to rapidly respond to the same pathogen if encountered in the future. In chronic viral infection or in cancer, the defense mechanisms are not effective or do not lead to complete elimination of the pathogen or cancer cells, leading to repeated release of antigen and activation of T cells, leading to their functional exhaustions. Exhausted T cells, therefore, arise from chronic exposure to cancer antigens and display sustained and elevated expression of inhibitory molecules such as PD-1, LAG3, and TIM3. They also fail to proliferate and produce antiviral and anti-cancer molecules. Amongst the most important molecules released by T cells is IL-2, interferon gamma, tumor necrosis factor alpha, granzymes, and perforins. IL-2 especially is fundamental to T cell biology, as it is required by cytotoxic T cells to perform their effector function, and interferon gamma, TNF-alpha, perforin, and granzymes, one released from T cells, can act directly or indirectly on different cells with the ultimate aim to kill cancer cells through their apoptosis. So all these cytokines are released from exhausted T cells at quantities that do not lead to effective cancer control. There is much interest in reversing or overcoming the state of exhaustion in the field of immunotherapy. So in order to assess efficacy of novel compounds, drug developers have been looking for robust in vitro assays using human cells that could recapitulate the most critical features of the exhausted T cells found in vivo. To address this market needs, we have established an in vitro model using human primary cells to generate exhausted T cells and demonstrated the ability of anti-PD-1 antibody to reverse the phenotype, leading to increased effector function and tumor cell killing. So on the slides that will follow, I will talk you through the assay setup and show some of the key results. So one of the biggest challenges in setting up this assay was to identify what in vitro methods and reagents we could use to mimic chronic exposure of T cells to tumor antigens. We based our approach on a method published by Balki et al. In 2011. So that's the paper referenced here. And to generate the exhausted T cells in vitro, we used conventional or resting naive T cells that we isolated from healthy donors as a starting point and subjected them to repeated rounds of stimulation with beads coated with antibodies triggering their T cell receptor and co-stimulatory molecule CD28, and in that way mimicking chronic antigenic challenge. We performed 3 rounds of stimulation, and at each round of stimulation, we assessed checkpoint molecule expression and secretion of cytokines to see if repeatedly stimulated T cells show indeed signs of diminishing effector functions with concomitant increase in checkpoint molecule expression. We used flow cytometry technique to measure expression of PD-1, LAG3, and TIM3 on unstimulated T cells, shown here in gray, T cells following one round of stimulation, shown here in blue, T cells undergoing 2 rounds of stimulation, shown in green, and T cells that have undergone 3 rounds of stimulation, shown in blue. We can see the changes on the histograms for the individual markers tested. The more the histograms are shifted to the right on the x-axis, the greater the marker expression. We can see that the expression of all markers is the greatest after 3 rounds of stimulation. We also quantified the signal and determined the percent of cells expressing all 3 markers. And what we found is that the cells start off as negative for either of the markers, and that through the subsequent rounds of stimulation, the frequencies of T cells expressing all 3 markers increases, so that after round 3, almost 50% of T cells express all 3 checkpoint molecules. We then wanted to see if repeated stimulation of T cells leads to impaired ability to produce inflammatory cytokines. We measured IL-2, interferon gamma, and TNF-alpha released into supernatants where activated T cells were cultured. So whilst T cells that were stimulated only once released copious amounts of all 3 cytokines, as you can see here, T cells that were stimulated twice or 3 times showed severely impaired cytokine release. So this really fits quite well with the diminishing effector function of repeatedly stimulated T cells. Next, we wanted to confirm in functional assays that our exhausted T cells indeed, when activated, show diminished responsiveness in comparison to non-exhausted T cells. We used a mixed leukocyte reaction, which is gold standard method for assessing T cell functionality, and we also tested T cells for their ability to kill tumor cells. In the MLR assays, T cells from one donor are mixed in with dendritic cells from a different donor, leading to T cell activation, proliferation, and cytokine release. For the tumor cell killing assay, we incubated T cells with tumor cells. In this case, we chose A549 lung cancer cell line and stimulated T cells through their TCR receptor and monitored viability of tumor cells over time in the IncuCyte machine. We can see that the non-exhausted T cells, when incubated with allogeneic dendritic cells, release high levels of interferon gamma and proliferate vigorously. Here are shown percentages proliferating CD4 and CD8 plus T cells, which are main subsets found within CD3 plus T cells. However, when exhausted T cells were assessed in the same system, interferon gamma release as well as T cell proliferation were significantly lower than that of non-exhausted T cells. We then measured the ability of exhausted and non-exhausted T cells to kill tumor cells. We tracked growth and apoptosis of A549 cancer cells over 7 days, either cultured alone, represented as gray line, or cultured in the presence of activated, non-exhausted blue line, or exhausted T cells, the orange line. You can observe that when A549 were cultured in the absence of T cells, they showed very few apoptotic cells. However, when activated non-exhausted T cells were added, the percent of apoptotic cells increased significantly, but that was not the case when exhausted T cells were used in the assays. The level of apoptosis in A549 cells was much lower in comparison to non-exhausted T cells. These results confirm that we have successfully generated human exhausted T cells. The next step was to evaluate if the T cell exhaustion can be reversed in the presence of anti-PD-1 antibody blocking the inhibitory PD-1 receptor, which is found in abundance on exhausted T cells. For this, yet again, we used mixed leukocyte reaction assay, but this time we added to the mix of T cells and enriching cells rising concentrations of anti-PD-1 antibody or a relevant isotype control. Again, we measured interferon gamma release and T cell proliferation of the two main T cell subsets. The dotted line on each graph represents the level of non-exhausted T cell activation. So you can see that the isotype control treated cells showed very low level of interferon gamma release and proliferation of T cells. However, in the presence of anti-PD-1 antibody, we notice a dose-dependent increase in all three assay outputs, confirming that indeed exhausted T cells can be reinvigorated by blocking inhibitory molecules expressed by them. Interestingly, something worth noting is that the level of reinvigoration never reached 100% of the responses observed with non-exhausted T cells, which suggests that there are multiple mechanisms at play that keep T cells in check, preventing them from reaching their full anti-cancer potential, and sets a suitable system for assessing potential combination approaches testing. Having demonstrated that anti-PD-1 treatment can partly reinvigorate exhausted T cells, we then sought to determine if there are other pathways that can be targeted in exhausted T cells in order to restore their functionality. So we looked quite carefully through the published data and identified IKZF3 as a potential target. This transcription factor has been shown to be increased in murine models of T cell exhaustion when using chronic viral infections. Activity of IKZF3 correlated with reduced expression of IL-2, and reduced expression of IL-2 is one of the hallmarks of exhausted T cells, and it was shown as well that it can reverse T cell exhaustion. So in order to block the activity of IKZF3 in the exhausted T cells, we used lenalidomide, which is a small molecule compound shown to induce enhanced T cell function and anti-tumor responses, but its ability to restore exhausted T cell function has not yet been reported. So lenalidomide can specifically mediate the connection between ubiquitin ligase complex and transcription factor encoded by IKZF1 and IKZF3, which are then ubiquitinated and degraded later, and then promote the secretion of IL-2 from T cells. So we hypothesized that lenalidomide can mediate increased degradation of IKZF3, thus leading to restored production and secretion of IL-2, which in turn can reverse T cells from becoming exhausted. We tested this hypothesis using our in vitro assay system for human exhausted T cells. So the exhausted T cells generated using our established protocol were cultured in the presence of allogeneic dendritic cells to drive their activation. The exhausted T cells showed reduced ability to respond to stimulation and exhibited reduced interferon gamma release and proliferation of the main T cell subsets. However, when lenalidomide was added to the culture, we observed dose-dependent increase in interferon gamma production as well as T cell proliferation. The reinvigoration, again, similar to as it was in the case of PD-1 treatment, was never full. So it was only partial reversal. So to summarize our data, we showed that exhausted T cells express elevated levels of PD-1, and they may also demonstrate increased activity of IKZF3, although we never formally demonstrated this. Engagement of PD-1 with its ligand PD-L1 found on dendritic cells provides an inhibitory signal to T cells, while AKZF3 suppresses production of IL-2, both pathways potentially contributing to an impaired ability of T cells to fight cancer. Monotherapy with nivolumab can block PD-1, PD-L1 interaction and partially restore activation of T cells. In a similar manner, monotherapy with lenalidomide can partly restore activation of T cells too. It will now be interesting to see if combined blockades of PD-1 and lenalidomide might be more effective in restoration of T cell function than either of the monotherapy used. So now switching gears to macrophages. This is another very prominent player within the tumor microenvironment. Indeed, tumor-associated macrophages are extremely abundant and are present in all types of tumors and constitutes, on average, 30 to 50% of stromal cells. Macrophages are phenotypically and functionally diverse population of cells. They are classified as two major subpopulations, including M1-like and M2-like macrophages. So usually the M1-like macrophages have anti-cancer effects, while M2 macrophages contribute to tumor progression. Macrophages are extremely plastic, meaning that they can switch from M1 to M2 and vice versa. And this unique ability of macrophages has been explored as a potential pathway for manipulation in cancer. TAMs, as they are often abbreviated, display a vast array of mechanisms that facilitate cancer persistence and growth. It is therefore extremely important to understand molecular mechanisms of TAMs and how best target TAMs in order to achieve clinical effects. So we have established a process to generate macrophages in vitro, which closely recapitulates TAMs. We used a method published by Carola Rees in her paper on CSF1 receptor blockades, which she published in 2014. So we differentiate M1 and M2 like macrophages from monocytes isolated from healthy donors, PBMCs, in the presence of either GM-CSF or MCSF, which are growth factors playing crucial roles in macrophage biology. When we differentiate M1 and M2 macrophages using the described methods, it is quite apparent, even when we look at the cells down the microscope. So both end products are very different from each other. So M1 macrophages show so-called fried egg morphology, whereas M2 macrophages show fibroblastoid appearance with very prominent vacuoles clearly visible. At the protein level, when we analyze M1 and M2 macrophages by flow cytometry, M2 macrophages, in contrast to M1 macrophages, show high levels of a surrogate TAM marker CD163, which is consistent with their in vivo phenotype. Indeed, CD163 has been, or CD163 expressing macrophages have been shown to correlate with poorer cancer prognosis in cancer patients in recently published paper. We have extensively characterized our in vitro differentiated M1 and M2 macrophages in terms of their gene expression profile, marker expression, and cytokine production. Shown on the slides are gene families frequently associated with macrophage biology. So at a quick glance, you can see how strikingly different M1 and M2 macrophages are with respect to gene expression. So just to pick a few examples, you can see that M1 macrophages express higher levels of IL-12 than M2 macrophages, whereas M2 macrophages express much more immune suppressive cytokine IL-10 in comparison to M1 macrophages. You can also see that both M1 and M2 macrophages express high amounts of PD-1 ligands, PD-L1 and PD-L2. And you can also see that CD206, unlike in mouse M2 macrophages, is not a good phenotypic marker for M2 macrophages, as it is expressed to the same extent or even in certain cases at increased levels in M1 macrophages. And CD204, which is another phagocytic marker, seems to be a better phenotypic marker on two macrophages in the human system. Macrophages in their natural environment are subjected to a variety of signals, either through released bacterial products or through damage-associated molecular pattern molecules derived from dying cancer cells. Many of these molecules signal through toll-like receptor 4, which is expressed on macrophages, both M1 and M2. We used lipopolysaccharides as a surrogate TLR4 ligand to assess cytokine secretion profile of M1 and M2 macrophages. We measured two cytokines which are crucial for shaping up T-cell mediated responses, namely IL-12 and IL-10. So IL-10 has pro-T-cell function, whereas IL-10 has anti-T-cell function. So we can see here that unstimulated macrophages release negligible levels of IL-12 and IL-10. But once they were activated with LPS, M1 and M2 macrophages show highly differential IL-12, IL-10 production pattern. So IL-12 was produced by M1, but not M2 macrophages, whereas IL-10 was produced predominantly by M2 macrophages and to much lesser extent by M1 macrophages. And this cytokine release pattern fits very well with M1 and M2 in vivo biology, M1 macrophages being pro-inflammatory, whereas M2 being anti-inflammatory. We use this simple monoculture assay to assess ability of different test compounds to reprogram M2 macrophages away from their anti-inflammatory cytokine signature. We briefly pre-incubated M2 macrophages, in this case, P38 MAP kinase pathway inhibitor, followed by overnight LPS stimulation. And the next day we measured IL-10 and IL-12 cytokine released into culture supernatant. We can see that the small molecule inhibitor showed ability to downregulate IL-10 release, and at the same time also upregulated IL-12 release from M2 macrophages, making them behave more like M1 macrophages with dampened down ability to suppress T cell function and with upregulated or enhanced ability to activate T cells. M1 and M2 macrophages have been shown to support or suppress T cell activation in vivo, respectively. We recapitulated these findings in our in vitro assay. So we co-cultured M1 or M2 macrophages with activated T cells and assess T cell function as measured by interferon gamma release at the end of the assay. We showed that when T cells are co-cultured with M1 macrophages, their activation and interferon gamma release is enhanced. But when T cells are co-cultured with M2 macrophages, their function and interferon gamma release is suppressed in comparison to T cells cultured in the absence of any type of macrophages. We next wanted to determine if we can block M2 macrophages from exerting their suppressive function towards T cells. M2 macrophages express high levels of PD-1 ligands, PDL1 and PDL2 at steady state, but they express PDL1 even more strongly following LPS activation. So these two ligands might interact with PD-1 expressed on T cells to switch the T cells off. We tested if blocking of PD-1 pathway within this assay could reverse the M2-mediated suppression of T cells. So indeed, when we added anti-PD-1 antibody to the M2 T cell co-cultures, we observed significant, although partial, restoration of T cell activation, very similar to what we noticed in the exhausted T cell assay. So yet again, the reason for it might be the presence of additional pathways secreted by macrophages that can dampen down T cell activation. To then evaluate, we used a candidate small molecule drug that targets M2 macrophages and evaluate it either singly or using 3 different concentrations or in combination with anti-PD-1 antibody. So as shown in white color bars, addition of small molecule drug reverse T cell expression in a dose-dependent manner, but the reversal was further enhanced upon a combined treatment with anti-PD-1 antibody, as shown in purple colored bars. I have shown that we have established a robust system to monitor differentiation and function of human M1 and M2 macrophages in vitro, and that we can reliably screen compounds for their ability to reprogram M2 macrophages and reduce their negative impacts on anti-cancer T cell responses. There are, however, multiple other approaches targeting tumor-associated macrophages, which are being looked at, and those include, as you can see on the snippets from the one of the latest reviews, promoting phagocytosis of TAMs to tumor cells, depleting M2-like TAMs with drugs by promoting TAMs apoptosis, and blocking the recruitment of TAMs to cancer cells. So I hope I was able to convince you that in vitro assay platforms using human primary cells are suitable to model complex cell-cell interactions which occur in the tumor microenvironment, and that these assays are useful tools to test potential new therapies to predict their efficacy in vivo. I would like to thank my colleagues for their inputs in establishing the assays and generating the results. I would like to thank you all for listening, and I will be happy to look into the Q&A box to see if there are any questions there waiting for me.
Thank you so much, Justina, for this great presentation with very practical information. And yes, as you said, now the floor is open for your question, please. And please type them in and send them to Justina or the pain list, then I will see them as well. Please do not type them in the chat, but in the question and answer section.
So we have a couple of questions. One of the question is, how many donors do we usually assess? And if we can see any donor-to-donor differences in response to compounds. So yeah, that's a good question. So most of the data that I showed you during my presentation was performed using between 3 to 6 donors, probably 6 donors. It's something that we would feel the most comfortable with when performing any human cell-based assays. The donors are obviously healthy when they are recruited to donate PBMCs, but they differ in their age, their gender, weight, whether they are smokers or not. However, we don't pre-select them based on this criteria. So some of the variation in the responses might come from these factors. The assays that I demonstrated to you, we don't observe great deal of donor variation. Whatever we observe might arise from some technical variation. So, for example, for the exhausted T-cell assay, we isolate T-cells from PBMCs, and then we subject them to rather strong, robust mode of stimulation in order to generate reproducible assay. So in that way, we provide that signal to the donors. Similar manner for our macrophage assays, we isolate monocytes and culture them in the presence of growth factors, and in that way differentiating rather uniform phenotype at the end of this differentiation process. So there is no huge degree of variation in these assays, and that can be, if there is any, that can be mitigated by using up to 6 different donors and the data at the end can be compared using statistical tools to inform if the differences between different treatment groups were statistically significant. So this is one of the question. Maybe one more question about PD-1 ligands, that you showed the expression of two PD-1 ligands on different cells, PD-L1 and PD-L2. What's the difference between them? Correct. So as I showed on one of the slides, PD-1 obviously being the major target in immunotherapy, it has two binding partners, PD-L1 and PD-L2. There are differences in engagement of PD-1 with these two different ligands. PD-L2 seems to have higher affinity to PD-1 than PD-L1. And as well, the expression pattern is different. So PD-L1 is expressed on different type of cells, not only immune cells, but also cancer cells, and it is inducible. So it's actually quite easy to drive PD-L1 expression on different type of cells, as well as on cancer cells, by simply treating them with interferon gamma. PD-L2, on the other hand, seems to be restricted to immune cells. And you might remember the gene expression profiles that I showed. It was clear that PD-L2 is much strongly expressed on M2 macrophages. Now, there are clinical antibodies targeting PD-L1 ligands. To my knowledge, we are lacking some of the knowledge around PD-L2 as an immunotherapy target, but it's potentially interesting target too, and worth exploration. So I think these are the question.
Yeah, I don't see any other question. So then thank you again very much, Justina, for taking your time for us this very presentation. And thank you all for your attendance. And I would like to let you know that if you exit this webinar, that there will be a very short survey with 3-4 questions. And please take a minute to answer this so that we may continue to improve our program. And Justina, back to you for a close of today's webinar.
So I would like to close this meeting, but obviously thanking you all for your participation, contribution, and interest in this extremely fascinating topic. Please stay in touch with us. Feel free to get in touch if you had any questions. Thank you again. I wish you a great day or great evening, depending in what time zone you are. So thank you very much.
Thank you and goodbye, and have a good day, and hope to see you soon in another webinar from Malvern Panalytical. Thank you very much. Bye-bye.
Bye-bye.